Hi, this is Shamin here. I'm from EducatorSharmin YouTube channel, Facebook page, Instagram and Twitter I have, so you can follow me there. So in this video, I'm going to teach you biology chapter 3, that is enzyme. So in this video, I'm going to teach you what is enzyme, how its work, and that is the factors that affect enzyme activity and Vmax and Km to compare the affinity of different enzymes for these substrates. And also I will explain how reversible inhibitors affect the rate of enzyme activity, and also carry out experiments with enzymes under control conditions. And also I will explain what's the advantage of using immobilized enzyme. Alright, so let's get started. What are enzymes? What are enzymes? So enzymes, that is our biological molecule, that is the protein molecule, which can be defined as a biological catalyst. So made of protein. Made of what? Made of protein. So that is they alter the rate of chemical reaction and without themselves chemically change at the end of the reaction. So a catalyst, what is catalyst? So catalyst is a substance which can speed up chemical reaction without itself being chemically changed at the end of the reaction. So it's a molecule which speed up a chemical reaction but remain unchanged at the end of the reaction. So that is a catalyst. For example, hydrogen peroxide is a poisonous in cell, but the cell produce the enzyme, that is the enzyme, that is catalase, which catalyzes the breakdown of hydrogen peroxide to water and oxygen. What's that? That is breakdown water and oxygen. What's the characteristics of enzyme? What's the function of enzyme? Number one, that is speed up chemical reaction. Number two, that is highly specific and digest food substance. That means whenever you eat something, is digest. Enzyme helps to digest your food and break down food substance in cells to release energy, and bind up, that is synthesize complex substance, for example proteins from amino acids in cells, and also control the numerous reactions in an organism. Now I'm going to talk about the lock and key hypothesis. So what is lock and key hypothesis? So the enzyme action depends on its active site. What is it? That is active site. So these sites are depressions on the surface of an enzyme molecule into which the substrate molecule can fit, just like a lock and key. Lock and key, just like a lock and key. So the enzyme substrate complex brings about the necessary reaction and converting the substrate molecule into product molecule upon substrate binding the product molecule and separates, leaving the enzyme molecule unchanged, and they are free to combine again. So now I'm going to ask you one question. What is active site? So an area on an enzyme molecule where the substrate can bind, and the idea that active site, the shape of the active site allows the substrate to fit perfectly. The idea that the enzyme has a particular shape into which the substrate fits exactly is known as the lock and key hypothesis. So what is substrate? What is substrate? So substance on which the enzymes act are called substrate. For example, starch, protein, fats. So substrate is the key whose shape that is the fits the lock of the enzyme. Talk about what's the difference between Vmax and Km. So Vmax, that is the theoretical maximum rate of an enzyme control reaction and obtained when all the active sites are occupied. So Vmax, that is gives information about the maximum rate of reaction, that is possible. So the higher the affinity, the more likely the product will be formed when a substrate molecule enters the active site, and that is the rather than the substrate simply leaving the active site again and before a reaction takes place. So here is a Michaelis-Menten constant, that is Km, the substrate concentration at which an enzyme works at a half its maximum rate, that is half Vmax. So used as a measure of the efficiency of an enzyme. The lower the value of Km, the more efficient the enzyme, and quicker the reaction will proceed to its maximum rate. So the value of Km for a protein, that is the potential enzyme, can vary and depending on a number of factors. Guys, this is a substrate, that is key, and this is the active site, and this is an enzyme. So here is an enzyme is a lock. So an enzyme has a cleft in its surface, that is called active site, and substrate molecule has a completely complementary shape. Substrate, this is a substrate that is has a complementary shape, and this is an enzyme substrate complex. That means a random movement of an enzyme and a substrate brings and substrates into the active site. So what is the active site? This is an active site and this is a substrate. So an enzyme substrate complex is temporarily formed. The R group of the amino acids and active site, that is the interact with substrate. So now this is a products, all right. So this is a products, and this is interaction of the substrate with the active site and breaks the substrate apart. An enzyme product complex is briefly formed. So before the two products, here is the two products, here is one, here is another one. That is molecules leave the active site. This is the active site, just leave here, all right. So here is another diagram, that is the enzyme, that is the lock, and this is a substrate, that is key. And here is an enzyme substrate complex, and this is enzyme and product complex. And here is an enzyme and this is a product. So this is a simplified diagram of enzyme function, how it's work. So you need to draw some diagram and color it. It can help you to remember. So this is enzyme and this is a substrate, enzyme and this is a product, all right. So lysosome breaking a polysaccharide chain. So this is a hydrolysis reaction. So what do you mean by hydrolysis reaction? So hydrolysis is a reaction that is a complex molecule to break down to simpler ones and involving the addition of water and tertiary structure of compact structure of a protein molecule and resulting from the three-dimensional coiling of the already, that is the coiled up. So here is an enzyme substrate complex here. So this diagram, that is showing that a space filling model, and here is an enzyme product complex. So here is another diagram, that is this is an energy, all right. This level is energy level, and this is a progress of reaction. This is a products and this is a substrate. So to change a substrate into a product, the energy of the substrate must be briefly raised by an amount of the activation energy. What is the activation energy? This is activation energy, and this can be done by heating the substrate. And here is another diagram. So here is another diagram, that is the energy level, and this is a progress of reaction, and this is a products and substrate, this line, dot line, and this is the activation energy. So activation energy, this is this diagram for with enzyme, and this diagram, that is without enzyme. What's the difference between with enzyme? This is a with enzyme, and this one is a without enzyme. So with enzyme, when a substrate bind to active site, so what is the active site? So active site of an enzyme, that is the shape of its molecule, is slightly changed, and it makes it easier to change the substrate into a product. That is the activation energy is lower. So this is a graph. That graph represents the effect of substrate concentration, and this is the initial rate of reaction, and here is a Vmax. So this is, you know, the Vmax, that is the theoretical maximum rate of an enzyme, that is controlled reaction obtained when all the active sites are occupied, and this is a substrate concentration. So the effect of substrate concentration, that is on the rate of enzyme catalyzed reaction. So if more substrate is added, the enzyme simply cannot work faster. Substrate molecules are effectively, that is the quicking up for an active site to become vacant. The enzyme is working at its maximum possible rate, that is, what's that? That is Vmax. And here is a V, this is a stand for, V is a stand for velocity, and max is a maximum. So this is another diagram, that is the effect of temperature on the rate of an enzyme control reaction. So this is enzyme becoming denatured, and this is optimum temperature rate, and that is 40 degrees Celsius. So optimum temperature rate, that is the temperature at which an enzyme catalyzes a reaction at the maximum rate, that is called optimum temperature, that is 40 degrees Celsius, and that is enzyme completely denatured here. This is a completely denatured, and rate of reaction, that is, this is go, here is a temperature rises, and here is a becoming denatured here, that is 40 degrees, that is optimum temperature. So here is another diagram, that is, you need to draw some diagram. It can help you guys to remember and understand the topics. So here, so this is a competitive inhibition. So here is the active site, right? And this is a substrate fields, that is the feeds precisely into the enzyme's active site, and that is competitive inhibitor has a similar shape to the substrate and fits into the enzyme's active site. So this is non-competitive inhibition. Other molecules may bind and elsewhere on the enzyme and destroying its active site. So this is active site is destroying its active site. So this is non-competitive inhibition. That is, the other molecules may bind elsewhere on the enzyme and destroying its active site. This is active site, and this is non-competitive inhibitor, and this is enzyme. So enzyme, that is that is the difference between competitive and non-competitive inhibitions. So here is another graph, that is the initial rate, that is half Vmax. Vmax, this is, you know, that is the maximum value, and here is a Km, that is the substrate concentration. So Michaelis Menten constant is the substrate concentration at which an enzyme works at a half its maximum rate of Vmax, that is the half, that is, if you see that diagram, you see properly, right? So Michaelis Menten constant, that is the substrate concentration at the which an enzyme works at a half, and its a maximum rate, that is the half Vmax, and used as a measure of the efficiency of an enzyme. That is, the lower the value of Km, the rate of efficient in the enzyme. So this is a diagram, that is the mixture of sodium alignate solution and lactase. Two things here. What's that? That is sodium alignate and lactase. This is, and here that is the one small drops of mixture enter calcium chloride. What's that? That is calcium chloride solution. So they form beads, and the alignate holds the enzyme molecules in the beads. So what's that? That is alignate. So enzyme lactase. So enzyme lactase here can be immobilized and using alignate beds, and milk is then allowed to run through the column of lactase containing beds. So lactase hydrolyzes the lactase is the that is the milk to glucose, and another one was that is galactose. So here this is a milk and this is alignate beds containing immobilized lactase, and here is a milk free of lactase and lactose and lactase, two things, that is lactose and lactase. So this is an immobilized enzyme to modify, that is modify milk. So let's review the chapter again. So here is a question number 1. The diagram below shows an enzyme and two inhibitors of the enzyme, that is X and Y, and which of the following describes the two inhibitors? So here is a diagram. You see that is, here is a X, this one is a X, and this is a Y, and this is a active site. So this diagram below shows an enzyme and two inhibitors of the enzyme, that is X and Y. X and Y, and which of the following describes the two inhibitors? What's the answer, guys? So here is the answer is B, that is X and Y are non-competitive inhibitors. Question number 2, that is to reaction controlled by an enzyme. Which of the following graphs shows the effect of substrate concentration on the rate of reaction? So if you see here, this is a graph here. This is 1, 2, 3, and this is a 4. So in a reaction control of an enzyme, which of the following graphs shows the effect of substrate concentration on the rate of the reaction? What do you think? Question number 3, that is the graph shows the progress of the digestion of starch by the enzyme salivary amylase. So why does the reaction slow down? Why does the reaction slow down? What's the reason? So his answer is D, that is there are fewer and fewer substrate molecules left to bind with the salivary amylase. Question number 4, that is if methylene blue dye is added to a suspension of yeast cell, living cells do not take up the stain and they remain colorless. However, dead cells are stained blue. So this fact was used to carry out an investigation into the rate at which yeast cells were killed at two different temperatures. At high temperature, the yeast enzymes will be denatured. The results are shown in the diagram. So this is a diagram, guys. So which of the following is correct? The higher temperature is the vertical axis, that is y axis should be level. So here is the answer is C, that is X, that is the person color cell. So guys, I hope that's all for today. Thank you for watching and see you soon with another new video. Keep watching and keep practicing. That way, draw a diagram, level it can help you guys. Thank you so much and good luck your biology test. Thank you so much.